Our protocol is significant because it uses the well-established technique of microinjection in Drosophila embryos to allow new research on the actin stress response and accompanying actin rod assembly. The main advantage of this technique is that we can study the actin stress response under a variety of contexts, such as a mutant screen or under different stress conditions. An individual who has never performed this technique before may struggle with handling the embryos and performing the microinjection. We recommend practicing these steps until the user feels comfortable. Visual demonstration of this method is critical because the steps of embryo handling, positioning, and mounting for both microinjection and imaging might not be clear from written instructions alone.
Prepare embryo collection cups and apple juice agar plates according to manuscript directions. To promote the most generous egg laying, set up collection cups with flies 2 days prior to the experiment. Add at least 100 female and 50 male flies to each collection cup, then cover it with an apple juice plate. Prepare a working solution of G-actin red and store it on ice until it is loaded into the microneedle. Allow flies to lay embryos for 30 minutes on the apple juice plates with yeast paste at 18 degrees Celsius. While the flies are laying, cut out a 4 by 1 centimeter rectangular wedge of apple juice agar with a razor blade and place it on a 25 by 75 millimeter glass slide. Harvest the plate from the collection and decorionate the embryos by pouring fresh bleach solution onto the plate and swirling it for 1 minute. Dampen the bristles of a paintbrush with distilled water, then use it to transfer decoration.